This is a trial with a new target called angiopoietinlike 3 and a relatively new molecule. And the target is a protein that's expressed and inhibits lipoprotein lipase activity. So if you look at patients that have loss of function mutations or complete loss of function in that protein, they have actually very low lipid levels. They have very low LDL, very low HDL, very low triglycerides. And this was actually noticed in a little town in Italy called Campo de Mele, which means Valley of the Apples, where there's a large number of these patients that are homozygous. So that really, along with some other data in animal models showing that actually can inhibit steatosis of the liver, makes it a very viable target for lowering triglycerides, LDL cholesterol, and perhaps having some effects on liver fat. So the study is using a drug to target angiopoietinlike 3, which is made in the hepatocyte. And it's using an antisense oligonucleotide, which is an RNA therapeutic, which is a relatively new field. And it uses some chemistry that allows enhanced uptake in the liver to have high potency and good tolerability. So this is the study that we did. This was a phase 1/2A study. It had normal volunteers that were given single doses, and they had volunteers that had high triglycerides where otherwise were relatively normal, or healthy I should say. So they were given different doses, and the dose effect was evaluated in that study. The findings were that we had a marked reduction in triglycerides and VLDL cholesterol. In the highest doses, about 60 to 70%. We had a reduction in LDL cholesterol and ApoB, as well as non-HDL cholesterol that were somewhere between 30 and 40%. And importantly, we had a reduction in apolipoprotein C3, which is another novel genetic marker of cardiovascular disease because it impacts triglyceride and triglyceride-rich lipoprotein metabolism. So the study showed very good safety and tolerability, and we're very excited about it because it'll address some unmet needs in patients that have residual elevated LDL for whatever reason, on top of standard therapy, elevated triglycerides. And it has a lot of potential for patients that have fatty liver, in which case we may be able to treat those patients with the drug because the drug looks like it may have some intrahepatic effects independent of its effects on triglycerides and LDL cholesterol. So we're just finishing up the last dose cohort for the follow-up. Once that's done, then our plan is to go to phase 2, which would likely involve cohorts of patients that have high LDL, high triglycerides, and those could be familial chylomicronemia patients, it could be patients with lipodystrophy, it could be patients that have high triglycerides that are not well controlled. And then we're going to look at the patients that have fatty liver and see if we can impact the amount of fat in the liver with this approach. This is probably a little too early to kind of think about cost. Cost is a complex issue. There's the cost of goods, and with this new version, we're able to give much lower doses than a prior molecule that we also tested in phase 1. So that should reduce the cost from that perspective, both in terms of the cost of goods. But cost is a complex scenario in terms of the market size and reimbursement. So I couldn't really give you any definitive answers because we're way too early to think about cost because we need to get through phase 2, and then we need to get through phase 3. Get FDA approval and regulatory approval, and then think about costs at that point, depending on what the population is.